We'll start with some more general questions that I believe have also been answered in your autobiography, like your interest in immunology and neurobiology. It's just mainly to set a precedent for future questions. So to start with, why did you choose to specialize in treatment of patients with multiple sclerosis? And why did you choose to specialize in fields such as neurobiology and immunology?
Right. Well, it's easy to answer. I can't think of an organ system since I went into medicine that's more interesting than the brain. I can't understand why people would want to become experts on anything else except the brain. I sort of smile, and I'm glad people go into fields like dermatology or kidneys or cardiology, because as I learned in medical school, they're all important. But the brain is the most fascinating organ. On a more personal level, I suppose that when I was very young, my sister got polio. So that was a devastating infection of her nervous system by a virus. So it became obvious to me that I wanted to study how the brain gets damaged either by a virus or in fighting the virus when it fights back. So I would trace it to that. When I was growing up, the family's heroes were Jonas Salk, who invented the polio vaccine. And I remember standing online at school, and we thought it was a miracle. Then through the same kind of ingenuity that both of you used to be at Stanford and studying these subjects, somehow when I was 17 years old, 18 years old, I got a job at the Salk Institute, not working with Jonas Salk, but working with some of the immunologists in his department. I was a physics major, so I chose to work with a physicist who converted his professional career to studying the immune response. And then how did I get to multiple sclerosis? Well, I decided to be a neurologist, to have a research lab. And my interests had at that time grown to both immunology and neuroscience. So the most common disease where the immune system is involved in attacking the nervous system is multiple sclerosis. But I'm interested in a lot of other neuroimmunological conditions. I think my career has been centered more on multiple sclerosis only because it's hundreds of times more common than some of the rare diseases I see. But as a matter of fact, there's an exceptionally rare disease where the current solution is to remove half of a person's brain. And I think we need to understand what triggers that disease and to come up with a better solution than removing half of a person's brain.
Certainly, yeah, that's very true. I definitely see the merit in that. Moving on, I was wondering, how long have you been in the field and treating patients specifically with multiple sclerosis, or looking for specifically researching the disease? And how many patients with multiple sclerosis do you tend to interact with on perhaps a weekly or monthly basis?
Sure. So after finishing medical school, I came to Stanford 50 years ago, 50, and started my clinical training after getting the MD. I interrupted it and I went to Israel for 3 years and studied immunology with an individual who invented one of the leading treatments for multiple sclerosis. Then I came back to Stanford in 1977, finished my 3 years of neurology training and joined the faculty in 1980. So my plan on July 1st, 1980 was to use my knowledge gained that far to try to understand multiple sclerosis, and maybe like the person I studied with in Israel, Michael Sella, S-E-L-A, I would also invent a new drug. So it's sort of shocking to me that most of the aspiration, or I could say all of the aspiration, has come true. I consider myself, as I wrote in the autobiography, very lucky. So many things can go wrong, and you can have high aspirations and never achieve what, when you were very young, you thought you might be able to do. It doesn't mean that I don't get up every day and run into walls or speed bumps or whatever you want to call them and say, oh, I'm not getting everything I hope to get. But I just have to keep laughing at myself for thinking that way.
That's fair. And actually, a follow-up to what you mentioned about Israel. I believe you mentioned you were studying a peptide-based drug, copolymer 1.
That's right.
What was your opinion on the effectiveness of copolymer 1 on MS?
Well, I think it had a benefit. If 100% is a cure, it had a benefit that's about 30%. So that's a good start. A couple of lessons were learned from the copolymer 1 experience. The scientists in Israel, in order to study multiple sclerosis, needed to get brain tissue. It's hard to get human brain tissue very easily, although we can get it. So they would go to the slaughterhouse to get cow brain. Most cow organs might, we eat a lot of, people who are not vegetarian eat a lot of cow muscle, otherwise known as steak or ribs. And to do that in Israel, they had to go through the rabbis and go through all the ritual slaughter practices that are part of being kosher. So they thought they'd be real smart and make synthetic myelin and see if that could cause an experimental disease. The experimental myelin did not ever cause disease. So somebody had the wise idea, well, maybe it will suppress disease caused by injecting experimental animals with myelin, and it did. So over a mere 25 years, it went from that idea to showing it more eloquently in animal models, mostly mice, and then they took it into the clinic where they saw some exciting results. So 25 years later, in 1996, the drug was first approved. And it was a big commercial success, even with that 30% efficacy that I'm talking about, over until it went off patent. The revenues from that drug were about $50 billion for a company called Teva. So it was really good for the patients, really good for the investors, and really good for the people who were the patent holders. The patent holders used their money very unselfishly. Ruth Arnon, who was one of the 3 patent holders on that invention, used the money to set up a whole campus at the Weizmann Institute for the Teaching of Science. So she lives in a very nice professor apartment. It's like a kibbutz, very small, very modest, but very, very pretty, and nice gardens around it. But she, instead of building a palace, she turned the money over to the institute where all the work was done, and they made a campus directed to teaching science, which I think is exceptionally cool.
That's certainly the case. Yeah. Thanks for talking about that. And I was wondering, jumping back to the research you do around current day, what would you say is currently the most effective approach for multiple sclerosis?
Well, we have about 15 approved drugs that have taken if the success of copolymer was to reduce flare-ups by about 30% in many of the people who took it. Now we're up to 60, 70, 80%. However, the treatments, and I've been involved in their development and inventing them, they're harsh. I call them big hammers. What I'd like in the ideal is to just remove the immune responses that we don't want to happen, the ones that attack the brain, and leave the rest of the immune system ready to fight viruses, COVID, polio, all of those things without knocking down the immune system severely. The good news is that the big hammers, the imprecise tools we have are very effective. As I said, it gets it up to 70, 80% efficacy from 30. And the side effects are consequential, but still very rare. So we're lucky. But I've spent a huge amount of time trying to find out exactly what we should suppress and to develop exquisite tools to do that. Unfortunately, we have some modest signals of efficacy. We've tried it in a few hundred patients, but it's nowhere near the big hammers. So the imprecise tools work. For instance, the one developed in my lab 30 years ago, the idea was, well, let's keep the immune system from even getting into the brain. And it works. The drug was approved, but within 3 months, the drug was pulled from the market because 3 patients died of a rare viral infection in their brain. Why did they get that infection in their brain? Because we blocked the immune system from entering the brain. So if the immune, it's like you have a neighborhood with a lot of gangs, and you tell the police, sorry, you can't get in. Do you have a time limit on your Zoom?
Oh no, that was just a notification for my webmail.
Okay, all right.
My bad.
Well, we're 1-to-1 because I had a phone call, you had a webmail. If I had one of those notification things, mine would be going all day long.
Oh yeah, that's true.
But wait until you get as old as I am. You'll be, maybe we'll have something else then. But anyway, if you stop the police from going into a dangerous neighborhood, the gangs would take over and make the neighborhood unlivable. So anyway, those are the balances. Perfection has not been achieved, and the imprecise therapeutics work very well with rather low frequency of serious complications. Now, when a person gets a serious complication, to them, it's horrible. And to me, as the treating doctor, it's horrible. But when I put somebody on these drugs, I tell them, we'll watch you, we'll take care of you. If we see something worrisome, we'll stop the drug and do something else. But it causes some degree of anxiety, obviously. You have a bad disease, you're offered a drug that can really slow it down or come close to stopping it. And yet the doctor says, and by the way, this could also kill you. It's tough, but sooner or later, most people face those kinds of decisions. Hopefully it's when you're very old, but the trouble with multiple sclerosis, it affects people generally when they're young, affects women more than men, and that's the horrendous part of the disease. You want to know, will I be okay? Will I be able to have a family? Will I be able to do all the things that I dreamed of doing? It's a tough one to answer, but with these drugs in hand, I can confidently tell them, we start the drug now, not start the more modest drugs and build up. We may be able to arrest the disease where it is, and you'll be able to, if you were a jogger, keep jogging. You won't end up in a wheelchair or in a hospital bed the rest of your life. And that's a tremendous change from the way things were certainly 50 years ago.
Yeah, that is a lot better. Actually, I'd like to ask a follow-up about the patients you mentioned. Since MS can impair vision and coordination, what options would you say are available to actually support these patients in activities of daily living?
Well, the idea is to stop the disease before there's impaired vision or impaired coordination. We don't have very good remedies for vision. If you can't do it by refraction, I've taken care of many individuals who really have severe impairments of vision, and it's horrendous. They used to be able to see, and now they barely see any kind of form. I think people in bioengineering may be able to help if there's, for instance, the circuit is shorting out, so to speak, in the nerves going to the brain. It may be possible to devise a chip that you could take the input going into the optic nerve and bypass the area of the short circuit. Unfortunately, that's an idealization, because the disease is patchy and it can occur elsewhere. And you brought up lack of coordination. That's an even bigger mess, because even if your muscles are strong, if I go to pick up a cup of coffee and I can't hold it without spilling it all over myself, what good is having the strength? So you want to arrest the disease before those things happen. But there is a lot going on in the area of neuroprostheses in order to help people who have these damage. It's primarily developed for people who have traumatic focal injuries. If you break your neck, will you be able to have something that allows you to move your arm? And I think that they're making significant progress. I read about it, and people very close to where I work at Stanford in engineering are developing great things. But multiple sclerosis is a bigger problem in a way than just getting a injury to your spinal cord or injury to one particular place, because then you could conceive of a bypass for that short circuit. Not so easy, but you can conceive of it. MS, much more difficult.
That's true. There are a lot of challenges, as you mentioned. And speaking of that, what are some of the biggest challenges that you'd say clinicians face in treating MS patients, if you had to name one?
Well, I think it's being able to predict who's going to have a horrible course and who's going to have a benign course. We have 15 drugs. Some of them are very potent. They're safer than I would have imagined, which is good. But I'd still like to be able to say, I've drawn your blood, I've looked at your scan, maybe I have a sample of spinal fluid, and based on the biomarkers, you have nothing to worry about. You'll be fine, even if I don't give you anything. That's the ideal. On the other hand, if we reach that state of affairs, which is a long sought after goal, what am I supposed to say to a person if their biomarker scan says that none of the 15 drugs are going to be very effective in you, and you're going to have a terrible course? Well, that presents a philosophical and ethical dilemma. You know, you don't want to lie to an individual, but you don't want to say something so pessimistic as to, you know, make them want to go into a depression or worse. So we face that with a lot of neurologic diseases. If I see somebody with amyotrophic lateral sclerosis, motor neuron disease, it's terrible. They're not going to do well. We don't have effective treatments, even though there's now a few conditionally approved drugs, or 2 are actually approved, but I only prescribe them so I can have something to prescribe. They don't work that well. Anyway, those are the kinds of dilemmas. Now, remember, 100 years ago, people didn't have all of these presumptions that they got to live to 70, 80, 90 years and face maybe Alzheimer's disease. All they had to worry about was escaping childhood infections and polio in the summer, et cetera, et cetera. So as we get better, our expectations get very, very, very high.
That's certainly true, and it's almost an optimistic outlook for the future. For the time being, do you believe that clinicians get sufficient support and training to effectively deal with the vast variety of MS patient cases?
I think so. You know, one of the privileges I have is I still see patients, and I still go on the ward and teach residents. They're so impressive. You know, I think they must be 10 times smarter than my cohort and I were when we were training half a century ago. Or maybe they know more because they work harder. But anyway, I think training, at least as I see it at Stanford, it's really exemplary. Also, they do protect people's time a little bit better than back in my days when, you know, it sounds like the person who says, when I was young, I used to have to walk to school and it was uphill both ways. That's of course, it can't be uphill both ways. So anyway, I think there's a field called topology where maybe there is an exception to that rule, but if you start at the same place, it can't be uphill both ways. Anyway, I think that training is a lot more humane, and the doctors that young doctors that I work with are exceptionally impressive. Another profound difference, when I was in medical school in a class of approximately 100, there were 5 women. Now it's in a class of 100, there will be about 55 women and 45 men.
True. I would also like to ask, because you mentioned before that it's very difficult to predict a horrible versus a benign course of MS. Would you say there's any additional gaps in understanding MS as a disease from like a clinical perspective?
Well, sure. One of the primary goals in my career has been to understand what causes MS. So in January of 2022, 2 papers came out. One, epidemiologic study done at the Harvard School of Public Health on 10 million blood samples from MS patients. And they showed really definitively with a p-value that had 4 zeros in front of it, that a virus called Epstein-Barr virus triggers multiple sclerosis. And our lab published a few days later the mechanism whereby there's a piece of that virus that has a resemblance to a component of myelin. So it got a tremendous amount of attention, and it's still absolutely thrilling. They named our work as a runner-up for Breakthrough of the Year for 2022 by Science Magazine. And people would come up to me and tell me, and you can tell I'm still laughing, we're so sorry that you only made runner-up. And I said, are you kidding? You know, first of all, it's a huge honor. Second of all, the first place winner was the James Webb Telescope. So, you know, like, it's okay to be runner-up to something that cool. But I think one of our gaps is to fully understand what's going on. And the more, this is true so often in science, that you make a discovery and then things get even more exciting because, to use the metaphor, you keep peeling different layers and you get deeper and deeper and it gets more complex. And what you thought was the answer is only the beginning of the answer. You know, there's a cliche, this is the end of the beginning, which means that you think you found something definitive, but now you find more and more fascinating paths. Anyway, I'd still like to exploit what we've come to understand to get to that perfect treatment for MS. But I'll tell you, at this stage, given the age on my driver's license, it does take decades to take a good idea and prove it in the clinic. So I won't see it, but I'm not sure that it can even be tested because the current drugs are so effective, because we have to look at it this way. If you came into my clinic and I could say, I could put you on this experimental treatment, which might be the cure, or I could give you an approved drug that is 80% of the way to the cure. I can't guarantee at all that my experimental drug will be the cure. What do you want? Well, the fact is that the ethical review boards will say you can never do that. What you have to do is put them on the approved therapies, the drugs that work 80%, and show that your drug can bring it up to 90 or 100. So to do that kind of experiment requires a huge group. If the effect you need has to overcome a huge barrier to get a little higher, you know, will 81% do it? I don't think so. You know, to get to, let's say, 90%, something that's perceptibly different, you might have to do a sample of individuals that's enormous. And who's going to pay for it? So, you know, we may be victims of our own success in preventing us from getting to the perfect answer. It's just the reality. You know, there are different fields of science where you don't have those realities, but if you have to test something in humans, and you have to have a p-value, statistical proof that you're better than the comparator, and the comparator is not a placebo, it's going to be one of the approved drugs. Good luck. It's going to take a lot of capital to do that.
That's really fair, yeah. And also, sorry, but I recall you mentioned like a while back that there were some hard-hitting treatments on the immune system that had about 70 to 80% efficacy. Do you think you could name them for noting purposes?
So we can start out with the drug that came from my lab. The generic name is natalizumab, Natalizumab. It's a monoclonal antibody directed to a Velcro-like molecule called an integrin, and it blocks the ability of immune cells to get into the brain. The drug is sold under the name Tysabri. It's been used by more than 250,000 people, and it's very effective. It has that downside that it could kill you, very rarely. But along the way, a risk mitigation test was developed. So 50% of people have a level of a marker that means they are highly, highly unlikely to get that complication from the virus that led to the drug being withdrawn before it was reinstated. The other class of drugs that works exceptionally well are drugs that kill the cells that lead to the formation of antibodies. So it's an anti-B cell therapy, and there are 3 approved anti-B cell therapies approved by the FDA. And they've been used at this point by about 1 million people. And they work as effectively, some people would argue more effectively, than the drug that targets the integrins. I was involved with the development of one of those 3 anti-B cell drugs, but they work, I put it at the 80% level. Some people would give it more credit. They say 90%, but it's not a cure, but it's darn good. Then there's some oral versions of those kinds of drugs that are not as effective as the intravenous versions of those drugs. But there's 15 approved drugs that include Copaxone, from way back. Although my opinion is, at this point in time, we have drugs that are so much better, it's not fair to the patient to give them the old drug.
Very fair. And Sergey, do you have any additional questions?
I think no. I think, Tanisha, you cover quite comprehensively all the kind of questions that we have prepared and thought about. And also, thank you so much to Professor Steinman for sharing the perspective and sharing with us the experience in the field. So really appreciate